Welcome to our second podcast on the urogenital system. And what we're going to do in this podcast is finish up just a small portion of our urinary tract, and then we'll move into describing the development of our genital tracts as well as the gonads and the genitalia. But first, I would like to show just a short animation of how we're going to divide this lower portion of our hindgut into the urinary versus the end of the alimentary canal. And that all-important structure that's going to do this is called the urogenital septum. In the beginning, we have the cloacal membrane, and this is going to be divided into two parts by the urorectal septum. And you can see as this grows down toward the cloacal membrane, we're dividing the very end portion of the hindgut into a part associated with urogenital sinus, and this is going to be part of our urogenital system, and the more dorsal part associated with our alimentary canal, which will end up being our rectum. Here you can see we've effectively divided these into the anal membrane and the urogenital membrane. As we return to the end of our urinary system, we covered the urethra. Now in males, this is going to transport both urinary and genital products. But what we see is that mostly the urethra is going to be comprised, the epithelium at least, of endoderm. In males, when we get to the very end, where we get to the glans penis, we're going to see that there's a portion of that, however, the plate is produced by the ectoderm. Now, these are going to fuse and they'll become part of the line system that we'll talk about. However, as we've seen in other areas of our alimentary canal, connective tissue, the smooth muscle, the collagens, these are all derived from splanchnic mesoderm. Now, as we move into the genital system, we're first going to look at gonadal development. And early on, there is an indifferent period of gonad development where you will not distinguish male from female. And so in these indifferent gonads, we're going to see they're derived from mesothelium of the posterior body wall, where the gonads are going to begin to develop, underlying mesenchymal cells, again, mesodermal in origin, and then primordial germ cells that are going to migrate in from the yolk sac. So these are going to be endodermal contributions to our genital system, and they're going to give rise to our oogonia and our spermatogonia. So here we are in the 5th week of development, and again, along the dorsal body wall, we have our urogenital ridge, and then on the very top, we are going to have the gonadal ridge that's going to form on top of that. It's sort of on, they say, the medial side. I think this is almost ventral in nature. But again, it's going to be a ribbon of structure that is going to form along the length. Now, in this illustration, they're trying to show you where this gonadal ridge is going to form. Again, this is somewhat in the lumbar region. But they're also illustrating this endodermal primordial tissue migrating up the yolk stalk and the umbilicus into the gonadal area, and these are our primordial germ cells that you can see migrating in. So in our indifferent gonad, we have epithelial cords, they're also called gonadal cords. They're going to grow down into the mesenchymal cells. So this is our underlying mesenchyme, and this is going to be our gonadal cord, often referred to as the sex cords or primary sex cords, diving down into this underlying mesodermal tissue. Now what this is going to create is somewhat of cortical tissue derived of the gonadal ridge, and the medullary portion, which is comprised of the underlying mesenchyme, part of the urogenital ridge tissue. Now this is sort of a generalized view, but it pretty much holds true as we follow through the development. In females, the cortex is going to become the majority of what we know as the ovary. The medulla, by and large, is going to degenerate. And it's just the opposite in males. Males, the medulla is going to become the bulk of the testes, whereas the cortex is predominantly going to generate. And so we'll see in our gonadal development, also our genital tracts, this is going to hold true. We can just expect the opposite happening in males as it does in females. Now those endodermal germ cells that we mentioned, they're going to begin to be visible about the 4th week, migrate in from the yolk sac. Following that yolk stalk, they're migrating in along that dorsal mesentery from the hindgut, and they will enter into our developing gonadal tissue. Now this is going to be complete about the 6th week of development, and these cells are going to be incorporated into those gonadal cords, which is also known as the sex cords. From the indifferent gonad, we're going to see that various hormones control the development either through a male pathway or a female pathway. And as we look at the development of the testes, an extremely early factor that begins to be produced is TDF, testis determining factor. Now, the default pathway for gender development is femaleness. So if this signal is not sent, if it is not detected, which means you have to produce the factor and have the receptor for the factor, then you will have female gender-specific tissues and organs develop. But in the presence of testis determining factor, the gonadal cords, they're going to extend into the medulla, they're going to branch, and they're going to anastomose to form a network. And this network is going to form the reedy testis. Now we're also going to see that seminiferous cords, also testicular cords, are going to form, and these are going to connect with the gonadal cords, and they're going to connect with the surface epithelium. So these cords, as they extend down, they're going to form the seminiferous or the testicular cords. These are going to give rise to our seminiferous tubules. We're going to have straight portions of these that are called the tubuli recti that connect to the reedy testes. Then with our efferent ductules, which in fact come from our mesonephric tubules, those then transfer the sperm from the testes to the epididymis. So here you see our seminiferous tubules, have the straight portion of the tubuli recti, and then the reedy testes. So here we have our indifferent gonad. There is our testis determining factor that leads us down the male lineage. Here's our no testis determining factor, and we enter into the female gender specific. But here we can see we've got our gonadal cords that have extended down. The first portions of our gonadal cords, they're going to give rise to the tubuli recti in this position. Here we have our seminiferous cords or tubules at this point. We already have our primordial germ cells incorporated into those. And these are currently suspended back to the body wall through this mesentery tissue. We refer to this as the mesorchium. This is actually the mesonephric duct. Here we have mesonephric tubules, and these again are going to become our efferent ductules. So when we look at our indifferent gonad, we can see that we're going to separate the seminiferous tubules from the overlying cortical tissue, and that this capsule, this is going to become the tunica albuginea. And we're going to see something called the tunica albuginea also around the ovary. But here in this early development, this very tough type of white tissue, this is going to be one of the earliest diagnostic features that the indifferent gonad has now beginning to follow the pathway toward maleness. There we've already talked about the gonad, which in this case the testes, being a part of the mesonephros. It's still suspended back to this mesonephros through that mesentery called the mesorchium. Now we can see a little bit later in development, we have our seminiferous tubules. We're starting to see some of these cortical cords diving down. These are going to be septa that will divide the testes into certain lobules. Here you can see this anastomotic network. That's going to be our reedy testes. And here we can see these mesonephric tubules. In this case, that will be an efferent ductule, taking the sperm from the reedy testes and transferring it to what was the mesonephric duct. Now this is going to be portions of the epididymis. Now some of the cellular components that we have present, we can see interstitial cells of Leydig, and these interstitial cells, which are between the seminiferous tubules, these cells are going to be the ones that are producing testes, producing testosterone. Now, these cells will begin to do that around the 8th week of development. In addition to testosterone, they'll also be producing another androgen, androstenedione. And again, these are going to lead to that masculine development and the development of secondary male characteristics. Now, within the seminiferous tubules, we also have cells called Sertoli cells. These are nurse cells. They're going to help support the development of the spermatozoa. It's going to be important that they do this because the spermatozoa are going to be distinct from the tissue of the testes itself. If these were somehow in contact with the blood supply, instead of indirectly through the Sertoli cells, then your body would establish an immune response. So this is an immune isolated compartment. So in addition to nursing or supporting the developing spermatozoa, the Sertoli cells also make anti-Müllerian hormone. Now remember, I said that the default pathway for gender development is femaleness. Well, it's not enough to turn on the male pathway. We must also turn off the female pathway in males. We saw how the mesonephric duct is going to become the epididymis and portions of the vas deferens and the ejaculatory ducts. Well, there is a second duct that follows along with the mesonephric duct. It's called the paramesonephric duct, and this is going to give rise to the female genital tract, and so that we have to turn off. Now, another name for this paramesonephric duct is the Müllerian duct, and so that makes a little bit more sense as we talk about the anti-Müllerian hormone. We have to shut that down. Now, we're going to see in females, it's just the opposite. That paramesonephric duct, the Müllerian duct, it persists, and they shut down the mesonephric duct, which also is known as the Wolffian duct. So that is going to be the opposites that we see development between males and females. Now, the seminiferous tubules, they're going to remain solid cords until after puberty. Once you go through puberty, then we're going to develop the cells, distinguish the cells. The Sertoli cells, again, are going to be producing the anti-Müllerian hormone very early on, but they're going to continue to support the development of the spermatozoa. Our primordial germ cells are present. They are spermatogonia, and these are going to give rise to the different stages of spermatozoa development. We study those all those stages in our histology course. We've mentioned already the mesonephric tubules will become efferent ductules, may have 15 to 20 of those, and portions of the mesonephric duct we simply recycle and rename and call that the epididymis or the ductus epididymis. Now let's shift gears, move away from male development, and now we look at the development of the female gonad. And in this case, we're not going to really develop these until about the 10th week. And again, much like what we saw in males, this is going to be the opposite in females. So our gonadal cords, those primary sex cords, they're going to extend into the medulla just like we saw. And as they form the reedy testes, these are going to form these gonadal cords as well as the reedy ovarii, but for females, both of those degenerate. They're not going to receive the testis determining factor. They're present just in case they do, but in this case, that signal's not present, so they degenerate. So now what happens is you form a second set of cords. These secondary sex cords are cortical cords, and these are going to extend down into the underlying mesoderm, that mesenchymal tissue, and this is going to give rise to the bulk then of what we see with the ovary, whereas the medulla is where we got most of the testes tissue. Now again, germ cells are going to be incorporated in these same yolk sac endodermal cells migrating in, just like we saw gave rise to spermatogonia. Here they're going to give rise to oogonia. So oogonia are going to be a part of what we consider to be the primordial follicles that are present in these ovaries. This is going to be lead to the development of the egg, the oocyte, but they're going to be supported by follicular cells. They're surrounded by them. These cells will continue to proliferate and grow, and we can talk about ovulation and the process by which that maturation happens, but again, we do that in our histology course. So when you're born, you have about 2 million or so of these primary oocytes or primordial follicles that are present in the ovaries, and every month once you reach reproductive age, you're going to mature some of these until one ovulates. And typically that's going to happen on alternating ovaries, right side 1 month, left side the other month. And the developing follicles that do not ovulate degenerate. Now we do see we have a tunica albigenia. This is going to be a thin capsule. This is not as diagnostic as what we see in the male testes, much less pronounced here, much deeper. And the mesovarium, this is going to be the mesentery that suspends the ovary back to our formerly mesonephric tissue. Here we see in our illustration there's no TDF, so we continue to develop the female pathway. Our initial gonadal cords degenerate. This was the reedy ovarii. That's degenerating. And now we see these secondary cortical cords diving in. We can also see that we've got our primordial germ cells here ready to give rise to our oogonia. Here we see a little further development. Again, degenerating reedy ovarii. The gonadal cords are degenerating as well. And here we're going to form the bulk of our ovary from our cortical tissue. Now, we've already introduced a little bit the genital ducts. We talked about the mesonephric duct and how that's going to give rise to the epididymis, part of the vas deferens. It's also known as the Wolffian. The female is going to have a second duct that parallels the mesonephric. That's where we get this paramesonephric term, and that's called the Mullerian duct. So, in males and females, both begin to develop, but the one not being used degenerates by and large. So again, to reiterate some of what we have said already, in male genital ducts and glands, mesonephric tubules give rise to the efferent ductules, again 15 to 20 or so. The mesonephric duct is going to become part of the epididymis, while the most distal portion, the part closest back to the body, this is going to give rise to the ductus deferens, or as we know it, the vas deferens. Now, as we approach the urethra, we're going to see that at this very end, we're going to have sort of a dilation of our mesonephric duct. This is going to develop into the seminal vesicle. And then between the seminal vesicle and the urethra, this is the portion that we term the ejaculatory duct. So here in our illustration, we have our testes. We can see our efferent ductules. Again, this is from our mesonephric tubules. Here we have the epididymis. It's often broken down into the head, the tail. We get into the vas deferens. We re-enter the body through the inguinal canal. And here, as we come in, this is going to be our ejaculatory duct. And then we have this dilation branching off. That is our seminal vesicle. Female genital ducts, we've mentioned this a little bit, but to go through it already, we've got our paramesonephric ducts, both right and left, and these are going to give rise to our female genital tracts. Now, these are going to encompass essentially our fallopian tubes and our uterus. And so as we look at forming our fallopian tubes or uterine tubes, we're going to see these are the paired unfused portions of our paramesonephric ducts, both on the right and the left. Now, as these ducts approach the point where we're going to have the uterus, then they're going to fuse together into a Y-shaped structure. Imagine the two upper arms of the Y being our uterine tubes, and the leg of that Y, that's going to develop into a structure known as the uterovaginal primordium. As the name implies, this is going to give rise to the uterus and as well give rise to the vagina. Now, the endometrial stroma and the myometrium that we have present, stroma being a connective tissue. So connective tissue in the muscle of the uterus itself, this is going to be derived from splanchnic mesoderm. So here we see in our illustration, we have our indifferent gonads that are developing. We're rapidly converting these into our ovaries. Here we have our mesonephros that's degenerating. Here is our mesonephric or Wolffian duct that is degenerating. We also have our paramesonephric ducts. In this case, since we are following our female pathway, they will persist. And then here we have our uterovaginal primordium. It's this blue Y-shaped extension, and this is going to connect back with the lower portion and the urogenital portion of our hindgut. So we are in our urogenital sinus at this point. There's the phallus portion of the sinus. We've got the pelvic portion and the vesicular portion. And here we have the rectum. We've already separated this with our urorectal septum. And so we're going to focus on now this portion, our uterovaginal primordium, and its connection back to this portion of the urogenital sinus. Now, as we bring those two branches, if you will, of our uterovaginal primordium together, these are our paramesonephric ducts now that bound together, we're going to have a portion that is derived from our urogenital sinus, and we're going to develop that shortly. But this is going to be that paramesonephric duct material. And so as they're brought from the lateral portion and fuse along the midline, this is going to create the broad ligament. Now, these broad ligaments, as they're pulled in, they are going to create two pockets, if you will. And these peritoneal pockets are going to be the rectouterine pouch and the vesicouterine pouch. Think it's almost like the pouch that you see on a kangaroo, if I can give an analogy, sort of trying to help visualize what's going on here. And so let's look at this in our section. Here we have a sagittal view. We can see the lower pelvic portion of our fetus. Here we have our paramesonephric ducts, our uterine tubes. Here we have our uterovaginal primordium. There we've got this portion of our urogenital sinus. And if we look at these different sections, if we look at them in cross section, we start very high up. So we're going to look at level B. What we're going to see, since they're separated, we can see the mesonephros, we can see the paramesonephric duct, the degenerating mesonephric duct here, the Wolffian duct, and we've also got the ovary developing here on the medial portion, but you see how they're separated. Here's our dorsal mesentery and our foregut, urinary bladders already separated, but we've got this one common compartment. Now, as these paramesonephric ducts, as they fuse together along the midline, they're going to be bringing this mesentery with them. This is going to be our broad ligament that we're pulling in. And so as we do that, let's go step down to level C. You see we're at that fused portion. So now we can see our degenerating mesonephros. We have now fused our paramesonephric ducts together, degenerating mesonephric ducts. I know that sounds kind of the same. It's easier to say Müllerian and Wolffian. It really distinguishes those two. But you see we pull them together, and a little bit lower down, we can see it even better. There's our fused uterovaginal primordium. And now we have these pockets. Here's the rectouterine pocket, the more dorsal portion, and the vesicouterine pocket. So if you're down in this lower portion that you see, there's no way to get from this compartment, which is more ventral, into this compartment, which is more dorsal, unless you were to move up over this point where they're connected and then drop down into this pocket. You see how they create these two pockets? I mean, they're just anatomically distinct compartments created by this mesentery. Now, where we have the uterovaginal primordium, the end of the paramesonephric ducts, where we have that contacting the urogenital sinus, this is going to create the portions of the vaginal epithelium. This is going to be endodermally derived. And yet the fibromuscular wall of the vagina in this point is going to come from our splanchnic mesoderm and the mesenchyme in the immediate area. So the point on the inside of the urogenital sinus, where that uterovaginal primordium contacts, we're going to see an enlarged bulb that's going to form. And this is called the sinus tubercle. Now, the sinus tubercle is going to be an inductive center. And the sinus tubercle is going to induce the formation of the sinovaginal bulbs. Now, the sinovaginal bulbs are going to fuse together, and this is going to form a solid column of tissue called the vaginal plate. Now, as we've seen in our alimentary canal, this solid column of tissue is going to have the central portion of that degenerate and hollow out. We call that canalize. And this is going to leave the lumen then of the vaginal canal. So here we see that contact point. This is a sinovaginal bulb on the outside. And there you can see on the inside, they've done a little cutaway view. That is the sinus tubercle that's present there. Now in this image, we can see the vaginal plate, this core of tissue that's forming. This is endoderm because they've got it in yellow, part of the urogenital sinus. Here we have the blue from the paramesonephric duct, and we can see that this is a solid column, this vaginal plate, and the middle is going to hollow out to form the lumen of the vagina, but this will not open up. Part of this vaginal plate is going to remain intact, and that is going to be this outermost membrane. And this is the hymen. The hymen is the tissue of the vaginal plate that persists until the female becomes sexually active, and this is mechanically ruptured. So in this particular text, we can see embryonic structures in different gonad, cortex, and medulla. Don't worry about this gubernaculum. This is really referring to various membranes. Call them the ovarian ligament here, the gubernaculum testis. I believe these are what we now refer to mesorchium and the mesovarium. Here we see mesonephric tubules, efferent ductules, the epididymis, which is referred to in this case as the perididymis, the mesonephric duct, perinephric. So use this to help you understand and apply the embryonic structure with adult derivatives in male and in female. So far we've developed our gonad, distinguished it from the indifferent gonad into our ovary and testes. We have our distinct genital tracts that we follow. Now we're going to look at genital development, and in a similar way, we're going to start out with indifferent genitalia. It's indistinct from each other, and we're not going to begin to see differences until the 9th week and really until the 12th week. And typically, obstetricians are not going to really do an ultrasound to distinguish gender until 18 to 20 weeks so that it's distinct, it's large enough to really be able to visualize, but some, can do it 13, 14, 15 weeks, depending on how urgent the need is to be able to see it. If it can be justified medically as well to do an ultrasound. They're not going to do one that early just because you want to know the sex. So what we're going to see is this indifferent genitalia. It's essentially going to be this open urogenital membrane, this open slit that was formerly the cloaca. And what we're going to see is at this cloacal membrane, at the cranial end of that cloacal membrane, we're going to have a mesenchyme enlargement due to proliferation. And this is going to become the genital tubercle. And this is going to become the phallic portion of our genitalia. We're also going to have two structures. We're going to have labioscrotal swellings and urogenital folds that are going to develop on each side of the cloacal membrane, or we can really refer to this as the urogenital membrane because we're distinguishing that. Remember, we're going to have the urorectal septum diving down, dividing this cloacal membrane into the anal membrane, and really a more specific term is going to be the urogenital membrane. Medial, closest to this membrane, are going to be the urogenital folds. Lateral to those are going to be the labioscrotal swellings. So here we have our indifferent. It's indistinct. We can't distinguish one from another. One is early stage, one is later stage. So here we have the cloacal membrane. There we have our genital tubercle. And again, as the urorectal septum comes in and divides this off, here we're beginning to develop our perineum. So here we have our anal membrane that's forming. Here we have our urogenital membrane. The phallus is expanding. Again, we're going to have our urogenital folds on the inside, and we're going to have our labioscrotal swellings on the outside. So here we have labioscrotal swellings. They're calling these the urethral folds. I still refer to them as the urogenital folds. That's how we learn them. So the genital tubercle, this is going to become the phallic portion. The urogenital membrane, this is going to be within that urogenital groove. This, again, is going to be created because of those folds on the outside. There's really no groove stretch. Kind of like the primitive pit, remember. That was only present because of the node that was surrounding it. So we've got this urogenital groove covered by the membrane, and it's created because the sides are the urogenital folds. Now, this urogenital membrane is not going to persist very long. This is going to rupture at this point, and this is going to leave this urogenital orifice opening. Now, in females, this is going to remain open. And to me, the difference between females and males, I like to simplify things as much as possible as we're learning these. Essentially, we're going to utilize this same tissue to create the genitalia. Females are unzipped. Males are going to be zipped up. And hopefully that'll become clear as we look at the remainder of this lecture. But this opening that's going to be created once we rupture our urogenital membrane, this is a common orifice where we have the entry of the urethra, the entry of the vagina into this common area. And this is referred to as the vestibule of the vagina. Now, male external genitalia, we'll cover as we get to the remainder. We'll cover this in just a bit. But for male external genitalia, as we start to develop this, we're going to have those urogenital folds, those that are most medial. These are going to fuse along the ventral surface, and the phallic portion is going to become the glans penis. But here we are going to fuse these urogenital folds, and they are going to enlarge, forming the penis. And within this, we're going to form the spongy urethra. That's going to be the lining of our urethra surrounded by this erectile column of tissue that is called the corpus spongiosa. And we're not going to get into all that detail too terribly much. We cover that in histology course. Now, the lining of this urogenital groove then, these endodermal cells, these are going to form the urethral plate. It's going to form the urethra on the inside. But as our urogenital folds fold around, we're going to see on the ventral ectodermal surface, we're basically going to see a line. This ventral line that's going to mark the fusion of those urogenital folds, and that's going to be referred to as the penile raphe. So this is the process by which this opening, this urogenital groove, we zip up. And so here we have our urogenital folds, and they are going to zip up, forming the shaft of the penis. Here we can see the phallic portion that was there. That's going to form the head or the glans penis. We can see the yellow on the inside. That's going to be the urethral plate. That's going to form the epithelium of the urethra. Now, we still have this enlarging perineum separating the penis from the anal opening. Now, as we see when we return, we're going to see that our labioscrotal swellings, and hopefully the name is letting you know where we're going, we're going to start to zip that up as well into forming our scrotal tissue. This is going to be the compartment in which the testes descend from the abdomen through the inguinal canal and drop into the scrotum itself. So we said that the phallic portion in males becomes the head of the penis. It again is going to be surrounded by ectodermal tissue. This is going to be the prepuce or the foreskin. The labioscrotal swellings zip up, fuse along the midline, and form the labioscrotal swellings, form the scrotum. And again, we're going to have a ventral feature. It's going to be a raised line right along the midline of the scrotum, and this is called the scrotal raphe. It's like the zipper line, where these tissues zipped up to form the compartment to hold the testes. For female genitalia, it's much easier. It remains unzipped. So when we left, we had the urogenital folds, we had the labioscrotal swellings, we had the urogenital groove that was open because the membrane ruptured, giving access to the vaginal vestibule. And so in this case, the phallic portion is going to become the clitoris. The urogenital folds become the labia minora, and the labioscrotal swellings are going to become the labia majora. So that's female genitalia. So once again, zipped versus unzipped, that's the real distinction for what we have present in male versus female genitalia. So here we can see in female, this remains open. We have the labia majora on the outside, labia minora. Here we have the vestibule of the vagina. We have the urethral opening. We have the vaginal orifice that in this case is going to be covered by the hymen. We also have the anterior and the posterior labial commissures. This is where they are connected to each other, where they come together at this point. The phallic portion in female, this is going to become the clitoris. We can see that here, covered over by portions of the ectoderm of the labia majora. And then we're going to have sort of fatty tissue that's going to accumulate, and this is called the mons pubis. This is just above where you see the commissure of the labia majora.